Hi, my name is Marcel Westenberg, and this is presentation 1.4 of the Practibar course package. It's dealing with what is happening in the lab about DNA barcoding. What are you doing in the lab and how are you doing that? So the presentation goes about PCR amplification, the cycle sequence reaction after the amplification, the Sanger sequencing and the important base calling afterwards, and a little bit about Phred scores. So, what is happening typically in the lab about DNA barcoding? So, you start with the DNA extraction, so you need a specific kit for that. Then you perform the PCR amplification with a polymerase kit, and you perform that in a PCR machine. Then you visualize your PCR product on a biogelactophrase. You purify your PCR products with a specific kit, perform the cycle sequence reaction, and you do that in a PCR machine again, and then you purify your cycle sequence reaction, and then you put it on a Sanger sequencing machine, and finally you analyze the data. So what do you use? So you use DNA or RNA in case of viruses from one single specimen or strain. Because if you use mixtures of multiple specimens, this can lead to unreliable data or data that cannot be analyzed, because you get double peaks or double sequences in a mixture, and because of the generic nature of your amplification promise. And so you always, for instance, if you look to fungi and you amplify the ITS sequence and you still have some plant material in there, you also amplify the ITS sequence of the plant because of the generic nature of the primers. So in this case, you need pure cultures. So for phytoplasms or viruses, it's impossible, hardly impossible to purify them free of plant material. So in this case, specific primers are used. So PCR products serve as input for Sanger sequence reactions. So you only amplify a small fragment from the genome. And these are often multi-copy regions. For instance, CO1 from mitochondrial DNA, so you have multiple mitochondrial in one cell. ITS, nuclear DNA, so you have different copies of ITS, and MAT-K for chloroplasts. Also, there are more chloroplasts in one cell, so there are multiple copies. So the positive part of that is that you have increased sensitivity by using those loci, but there is also a downside, because you can have multiple haplotypes between different copies. So a little bit about PCR amplification. So here you have your target sequence, and by denaturing it at 95 degrees, you get 2 strands of your DNA. So by lowering the temperature to, for instance, here to 55 degrees, your primers anneals to both strands of the DNA. Then you have elongations with your magnesium chloride, your polymerase, and your DNTPs. So you elongate it from the primer to some. So what happens then after one cycle? You get those products right here. So you don't have your amplicon yet. So if you do another cycle of denaturing, binding primers, and elongation, you get 4 products and still not your amplicon, because that happens after cycle 3. You got for the first time your desired amplicon. And so you can do about 35 cycles. We got a lot of those products. You see here, 35 cycles give you a lot of amplicons. So everybody make mistakes, so also polymerases can make mistakes. Yeah, and so if errors are introduced in the early cycles, it can introduce erroneous bases and lead to wrong sequences. So it's very good to use proofreading enzymes when you're using DNA barcoding. So without proofreading, you can have mistakes of 1 into 10,000 bases, and with proofreading, it's about 1 into 10 to the 8 bases. So what is a proofreading enzyme doing? So for instance, here you see you have a wrongly incorporated G. Yeah, so you got a GG here, and proofreading enzymes has a 3 to 5 exonuclease activity. So it can get rid of this G again, and then can build in the right C. Yeah. So polymerases 5 years ago, which were proofreading enzyme, which are still sold right now. They don't call that proofreading anymore because the polymerase becomes better and better, and there are new polymerases on the market, coming on the market every time. Yeah. So, but they still are proofreading. Most of the enzymes are now proofreading enzymes. So how can you monitor possible introductions of read errors? That's already presented in another presentation, and that is because you can use positive amplification controls. For instance, you can use synthetic DNA constructs called G blocks. So they have a sequence, you know which they have, and they have the primer sites on it as well. So every time when you do an amplification, you also do the amplification on a G block. And by sequencing those amplicons, you can find out if there are errors introduced, because you know what the sequence should be. So it's always good to use amplification control. So PCR primers are self-fulfilling prophecies. Yeah, so amplification primers with mismatches that initiates elongation will give a perfect match after sequencing. So for instance, you see here a primer which has a mismatch right here, but it still can bind to the DNA, and so you get elongations. And when you use the opposite strand, later on, you see you get the sequence incorporated of your primer and not from the original sequence. So, for instance, I can show you in Genius an example. Like here, we have the CO1 sequence of Ampliflora glabripanus, which is a Coleoptera. So we have here the primer LCO1490, which is used for amplification of CO1 sequences of arthropods. And this primer has 4 mismatches to the sequence from the Ampliflora glabripanus. And also on the other side, it's the reverse primer that has 3 mismatches to the template. But we could still sequence them with those primers, and we could got very nice sequences, like you see here. And when you align those sequences, you see here the original sequence from the specimen, and here the trace files. So you see that in the trace files, you find back the primer sequences and not the sequence from your original specimen. So this is very important, and so you always need, when you make consensus sequences, you always need to trim your sequences so that the primer sites are trimmed from your consensus sequences, because you never know if that sequence is also in the original sequence of your specimen. So go back to the presentation. And we start with visualization and amplicon purification. So when you have, you have here your gel, and you see here you have a nice band, and so this band you can directly purify for a sequence. But if you have multiple bands, like you see here, it's important to gel purify both those amplicons from gel and sequence them separately, because those have different sequences, and if you sequence them directly, you get multiple peaks at the same position. So cycle sequence reactions is done as a linear amplification. It's basically a PCR with only one primer. And you need DNTPs and also labeled DDNTPs. So, and the difference between those are that a normal DNTPs has here the position 3 a OH group, and DDNTPs has a H group right here. And those DNTPs also has a fluorophore. But if this DDNTPs is built in, then the elongation stops, because it's not possible to add another NTP right here. So you do the cycle sequence reaction twice, so one with the forward primer and one with the reverse primer in separate reaction, so that you sequence it by direction. So what about sequence primers? So here you have your genomic template, and you can amplify with primers, and you get your amplicon, and you can sequence that with the same as your amplification primers. Another option is that you do an amplification with primers which are M13 tilt. So these are parts which don't anneal to your original DNA template, but they're built into the amplicon, and then you can sequence with M13 sequence primers. And then the third option is that you have amplification primers, that you sequence it with internal primers. So this is, for instance, done in the DNA barcoding protocol for bacteria for the 16S, where you amplify a very large product of 16S, but then that one is sequenced with 2 internal primers. So Sanger sequence detections, how does that work? So you have your sequence primers, DNTPs, and your labeled DDNTPs. And so when it starts, so you have your primer again, you build in different DNTPs, and then by chance a labeled DDNTPs is built in, and the elongation stops. So you get different fragments, different lengths, with different fluorophores in the end. And you separate those different fragments in size by using a capillary electrophoresis on the Sanger sequence machine. And so the smaller fragments comes off first, and in the end of the capillary, the fluorophore is detected. So you cut things like this, so different sizes and length. And first the T is coming off, then the C, another C, and A. And so you get your whole sequence. So a word about base calling. So different labels on DD NTPs migrate at different speeds. So your raw Sanger sequence data is a little bit messy. So you need to perform base calling later on in silico on a computer. So for instance, here you have your sequence before base calling. And so you see it's a little bit messy. After the base calling, you get nice sequences. So if you outsource your Sanger sequencing to another company, then this base calling is already done by the company. So you get your trace files, which are base called. So you don't have to do that yourself. So I want to show you an example that base calling can also go wrong. And that's because we go to Genius. And so for instance, here we have two different trace files. And so this trace files has some base calling done, but it doesn't look that nice. You see some broad peaks and overlapping peaks, so that's not nice. That's because there are two different kits we use in our laboratory. So that's the big die terminator kit version 1.1 and version 3.1 from Applied Biosystems. The version 1.1 is for short amplicons, and the 3.1 is for long amplicons. And you need different base calling settings depending on which kit you use. So here you have base calling done with the wrong settings. If you can correct that, so you can redo, so you can go back to the raw data which is still on the computer and redo the base calling, and then you can get nice sequences like this. So go back to the presentation again. So a little bit words about Phred scores. So Phred scores give a measure of the quality of a particular base. So for instance, a Phred score of 10 means that the 1 out of the 10 bases are probably incorrect called, and you get a calling accuracy of 90%. And so if you have a Phred score of 40, which is regarded as a high quality data, there you have a probability of incorrect base calling in 1 out of 10,000 and get an accuracy of 99.99%. So what makes the Phred scores? So the Phred scores is based on the peak shape. Yeah, we have a very high and sharp peak, then you get a high score. So here you have very nice sequence, and you can see here because of the color it's light blue, you have Phred score which are higher than 40. So this is a nice sequence. For instance, here you have a double peak, and so the Phred scores become lower. So it's here darker blue, so it's between 20 and 40. Here you have a peak height which is lower, and you also have a lower Phred score. But this lower Phred score, like you have here, you have an A, it's still nice. And you always have your other trace file from the reverse read. And if there is an A as well, then you get nice, still a nice consensus sequence. Here in this lower, it's very ugly because of dye blobs. You can here see, and then so you get very low quality trace files, bases with lower than a Phred score lower than 20. So the Phred score also decreases with length. Yeah, so you see always a low Phred score in the beginning, like a startup of your trace file. Then it becomes nice, with good quality, and then later on it's tailing off. Yeah. And it's influenced also by the different kits you use. Yeah. So if you have longer reads, then you use 3.1, then the tailing off start becomes later, but then the start up is also a little bit larger. And of course, in your forward read and your reverse read, you see the same thing. But if you assemble those, by using the bidirectional sequencing, then this poor region here is in the other side, so the other trace file has better or good quality, and so in the end, if you make consensus sequences, you get nicely sequences, with good quality. So therefore, it's important to do bidirectional sequencing. Okay, thanks for listening to and watching to this presentation.